Hey, so this is case study number 2 in our cBioPortal practical. So we're back here at the cancer type summary page, looking at the proportion of P53 mutations across the entire Cancer Genome Atlas cohort, these 32 different studies. Now, we've been focusing a lot on the big mutation frequencies, so things like esophageal or lung or head and neck cancers, because they have a lot of P53 mutations. Now, what I'm interested in actually is going right down to the bottom here, to these acute myeloid leukemias. So in these dataset, it's only about 9% of cases which have a mutation in AML. So let's have an explore and see what's going on with these mutations. So looking at the oncoprint, you can see there's very, very few. And if we go into the mutations plot, the lollipop plot, you can see that whilst the mutations are in the places that sort of make sense, there's not enough to actually find any kind of hotspot. So there's just mutations across the main domains. So there's very few mutations, but if you do have a mutation in P53, your survival is way, way worse. You can see your median survival here is only 5 months after diagnosis, compared to, and it's not much better, really, but compared to 17 months, so a year and a half in the unaltered group. So P53 mutations are much worse for survival. But not very many tumours have them. And actually, AML overall doesn't really have that many mutations. If you click here on the study at the top, open it in a new tab, this takes you to the full dashboard for this particular dataset, the Acute Myeloid Leukaemia TCGA dataset. Now you can see here that the number of mutations is typically less than 40. So the mode value being this kind of 5 to 10 range. So the number of mutations isn't very high at all. That would compare to something like bladder cancer, where it's hundreds to thousands of mutations in a sample. So what's going on here? Well, so if we go back to this plot again, we can see up here on the side that some of the other genes that are really commonly mutated, P53 is here, but it's much, much lower, are things like DNMT3A, so genes involved in methylation. But a key one I want to focus on here is this one called NPM1. So this is a gene that's mutated a lot in AML. So if we go to the edit box back on the AML only page, add in NPM1, and then submit the query. What you can see is that the altered group still has much worse survival, so a mutation in either, but it's not as worse as P53 alone. So we can untick the altered group here and look at P53 alone, so that terrible survival from the previous plot, or look at NPM1 alone. And whilst the survival is worse than unaltered, you can see it's still a bit better than in the P53 cases. So 5 months average median survival for P53 mutated tumours, about 10 months for an NPM1, and you're looking at kind of, now it's gone up because we've taken out those NPM1 tumours, you're looking at somewhere in the region of 2 years for the unaltered group. So let's have a look at some of these mutation plots. So you can see this is our P53 one again. NPM1's an interesting one because all the mutations happen right at this end here. These are actually the same site, it's just different types of mutations, different types of frame shift mutations. So frame shifts have a similar result to nonsense mutations. So here we've got insertions or deletions of bases, and that changes the reading frame, changes the ability to make a protein, causing it to truncate. Now, usually when you're talking about a truncated protein, you wouldn't think that a truncation right at the end matters too much. But actually truncating it here, this very, very C-terminal end of this protein, is really, really important for its function. Changes where it locates within the cell, and so it has a very different impact on what the cells are doing. If we look at the oncoprint, we can see that these are very, very exclusive. So there's no overlap at all, and we can see that with a significant p-value and q-value. So q-value is just a significance value that's been corrected for if you're going to do big numbers of tests. So these two mutations do not happen together. So do they happen to be in the same pathway like we looked at the P53 pathway before? And no is the answer. We don't actually, NPM1 doesn't really appear in some of the key cancer pathways that we would think about. So what's going on here? Well, the key difference here is the fact that NPM1 mutated tumours and P53 mutated tumours represent totally different subtypes of disease. So you can, in our last example, case study 1, you could hit lots of different parts of the P53 pathway to affect P53 activity. In this example, what we're showing is that NPM1 can be affected taking your tumour down one route, or P53 could be affected taking you down a totally different route. So in this scenario, we're showing two different routes that tumours can take to become tumours. Now, even more crucial than hitting the same pathway, this means that patients in these two groups need to be identified, need to be treated differently, and we can see they've got different survival. Their tumours are very different. So this is why things like sequencing and genetic profiling are entering the clinic, to show the differences between these two types of tumours. Okay, it's a brief case study. Thanks very much for listening.